All right, so we have Jen and Amy talk about how the tissue are generated and formed in this session. And as you can imagine, once the tissue get formed, it's not going just to stay in the same states forever. You know, the environment is changing, you know, such as the metabolic states and nutrition states, everything is constantly changing. So really the key question is that how the tissue can actually keep evolving to adapt to different states, depending on the environment that the tissue get exposed to. So we are very glad to have our last speaker of the session, Dr. Lucy O'Brien from Stanford University, to tell us something about such topic. So Dr. O'Brien, as I alluded to, is interested in the adaptive mechanism that enable tissues and organs to be flexibly controlled in response to changing physiological needs. Her unique approach is really looking to the cellular and molecular details that underlies the dynamics at the tissue and organ level over space and time. Dr. O'Brien, we are very excited and look forward to your talk. Please take over from that.
I want to add my voice to all the speakers and thanking How Long, you, Yuan, Ross, for organizing this amazing symposium. Fantastic talks, and I'm super excited to be here today. I'm going to talk about the factors that control the speed at which stem cell daughters differentiate. Now, we heard from Micha this morning about differentiating of developing myoblasts and the role of stress in that. I'm going to talk about differentiation of stem cell daughters in adult organs in vivo and the implications at different differentiation rates might have for quality control. Now, the Waddington landscape is undoubtedly very familiar to many of you in this audience. This was first articulated by Conrad Waddington in 1957 as a conceptualization of cellular differentiation. The idea is that this ball at the top of the hill is like an undifferentiated cell. And as a cell differentiates, it would be like the ball rolls down the hill, choosing between different valleys that represent different ultimate cellular fates. Now, the Waddington landscape, I'd say, is timeless in both senses of the word. It's timeless in that it's unaffected by the passage of time, perpetually relevant, valuable, and appealing. I mean, Waddington published this in 1957, and it's still a very useful conceptual framework today. But this landscape is also timeless in the other sense of the word, that is existing or operating outside of or without reference to time. A ball rolling down a hill, you know, that happens in a Newtonian universe, but there's no dimension of time in this Waddington landscape. And yet we know that when cells differentiate into terminal fates, this happens in the context of time. So does time matter in the Waddington landscape? Does it matter for cell differentiation? I'm going to argue today that it does. And I think that maybe an alternate analogy we want to consider, this might be because of my past life as a ski bum, but it would be the runs at a ski resort. So this map shows North Star Tahoe Resort in California. And I want to make the point here that you can have the same start point and the same finish point, but many different ways to get down the mountain. If the weather is sunny and clear, I might choose the scenic route down the mountain. I might take some selfies with friends, that at the Mid-Mountain Lodge for a cappuccino, and then finally end up at the base of the bottom. But if the weather is stormy and windy, I'm not going to take the scenic route down the hill. I'm just going to hightail it down the slope as fast as I can and get back to the lodge. So just like skiers might choose different ways down the mountain depending on what the weather is doing, I'm going to show you today that stem cell daughters have different rates of differentiation depending on what the tissue is doing. So in our lab, we think that the hands down best model organ for studying stem cell biology in vivo is inside this beautiful creature here, the adult fruit fly. Now, I'm talking about the fly midgut. And through Alan and Will's work at the Carnegie, I'll wager that many of you in the audience are fans of this little organ as well. Now, the fly gut, functionally equivalent to the vertebrate stomach and small intestine. It's lined by this beautiful epithelium here. Epithelium made up mostly of terminally differentiated cells called enterocytes. These enterocytes have these big blue polyploid nuclei, and they're outlined in red. And it's the enterocytes that are the workhorses for the gut's digestive function. Now, this epithelium was long thought to be post-mitotic, but then 15 years ago, there was a game-changing discovery by Ben Olstein and Allen at the Carnegie, by Craig McKelly and Norbert Peramon at Harvard. They showed that this is actually a stem cell-based organ undergoing continuous renewal. It was the first self-renewing somatic organ to be identified in adult Drosophila. I remember when these two papers came out, I was a new postdoc at the time, and I was just so excited about this opportunity it opened up to bring all the awesome genetic tools of Drosophila to look at basic questions about how stem cells and their terminal progeny work together to ensure organ form and function. So the stem cells are these little green diploid cells here. They're just scattered throughout the enterocytes. They're essentially the only cells in the gut that divide. And when they divide asymmetrically, they go through a transition stage called an enteroblast before terminally maturing into one of these fully differentiated enterocytes. And it's this absorptive lineage, the stem cell enteroblast enterocyte lineage, that I'm focusing on today. Now, recently, my lab developed the first long-term imaging of the midgut in situ. We do this by cutting a window inside the back of a living fly. This exposes the gut, and we can image directly through the window. And so with this arrangement, the fly continues to eat, digest food, and even poop, all while we're imaging its gut. And we can image the gut overnight and come back in the morning to a fly that's very much still alive. So here's an example of the kind of footage we get. This is 8 hours in the life of a midgut. The nuclei of the enterocytes are in cyan. The cytoplasms of the stem cells and enteroblasts are in yellow. And this tsunami that you see rippling through the gut, these are peristaltic contractions of the gut tube as the gut digested food. And so with this setup, we can watch stem cells divide, daughter cells differentiate, mature cells die and get sloughed off, all happening in real time in an organ that's physiologically functioning. Now, Erin Sanders was a new grad student in the lab when we were developing this imaging technique. And she was really taken with the notion that injury might change the speed at which a cell differentiates. And she wondered if this was something she could watch happen in real time. So here's one of Erin's movies of a healthy gut. This gut has live markers for the stem cells, enteroblasts, and enterocytes, the 3 stages of the absorptive lineage. Now, to injure guts, she feeds flies the toxin bleomycin. This is the DNA damaging agent that acts on the enterocytes. And here's a movie of one of these bleomycin damaged guts. Now, there's a lot going on here. The gut is clearly sick. There's cells moving all around. An occasional immune cell zips across the screen. But what we're focusing on today is how cells are transiting through these 3 cellular states. So here, I want to take a minute to go into some of the key details of this differentiation process. Like many other cell types in developing and mature tissues across metazoans, this fate decision involves Notch. Notch is the master regulator of enterocyte fate. This is beautifully shown by Ben and Allen. They demonstrated that stem cells express both the Notch receptor and its ligand delta. And when delta in one daughter activates Notch in another daughter, that turns the second daughter into an enteroblast. And this enteroblast at some point becomes committed to its terminal enterocyte fate. But the enteroblast is still immature. And then over time, it'll differentiate to become a fully mature enterocyte. And during this second phase, it loses Notch activation. And so we think of this Notch off process as a marker of maturation. So Erin really wanted to watch both stages of this process, commitment and maturation, live. And we were really lucky that Liha and Norbert Perrimon had just developed a great tool that would allow us to do exactly that. This tool is the Transtimer. It's a bicistronic cassette containing a fast-folding, fast-degrading GFP paired with a slow-folding, slow-degrading RFP. Erens combined this cassette with a sensitive notch response element. And so the idea here is that during commitment, when notch gets activated and goes into the nucleus, it drives transcription off this NRE, and so the Transtimer turns on. And we would expect to see this in cells by having a quick spike up of the fast-folding RFP, followed by a slower rise in the slow-folding RFP signal. Then during maturation, when notch turns off, the Transtimer would no longer be expressed. And here, we would expect to see the GFP signal quickly extinguish, since this is a fast-degrading GFP, followed by slower loss of the slow-degrading RFP signal. Okay, so this is all in theory. Now, what happens in real life? So here's Erens' window mount imaging of a notch-driven Transtimer in a healthy gut. We can see individual notch-activated cells here, and some look like they might be changing color. And here's Transtimer in an injured gut. Now, there's more notch-activated cells here overall. The cells tend to be bigger, and intriguingly, there are more cells in the injured gut that are red, but not green. In other words, notch is recently turned off in these cells. If we zoom in on single cells, we can find examples that are going through this green-red Transtimer cycle. So here in the course of this 20-hour movie, this cell here is turning notch on. We see the GFP appear, and then a split second later, the RFP appears. And then the cell turns notch off. You see the GFP start to fade, followed by slower fading out of the RFP. And so Erens quantifies this GFP and RFP fluorescence over time to get a profile of notch signaling kinetics in a single cell. And what's really satisfying is that a lot of these traces are actually not that far off from what we would predict in theory, like this one here, where we see the GFP go up, followed by RFP, and then the GFP decrease with slower decrease of RFP. But what I haven't told you so far is that this cell that we've been looking at, this cell comes from an injured gut. And Erens could find lots of cells like this in the injured guts, ones that are notch turns on and then turns off. But she had trouble finding cells like this in healthy guts. What's more typical instead in the healthy guts is that she can see one phase of these dynamics. For instance, here, one cell turning notch on over a 20-hour period, or keeping notch stable, or gradually turning it off. And so here, this is a composite of 4 different cells in the healthy gut to get this on-off cycle that we see in one cell in the injured gut. So, and in fact, Erens counts all these Transtimer cells. She finds that in injured guts, there's over 2.5 times more cells going through this on-off cycle compared to healthy guts. Whereas in healthy guts, there are more cells that show only the on phase or only the off phase. So this notch on-off cycle is more prevalent with injury. Could this be because the signaling amplitude of notch gets lowered when the tissue gets damaged? And so cells max out faster, the bar is lower for them to cross, and then they just go back down. Well, to get at this, Erens took movies in which she could identify all the stem cells in enteroblasts using an orthogonal marker, escargot. This histogram here shows levels of notch activity across this entire population of stem cells in enteroblasts. It's instantaneous GFP fluorescence on the x-axis. Now, the overall population distributions are different in injured and healthy guts. I'm happy to talk about that more in the questions. But the take home here is that the injured guts show the same upper range of notch activation as cells in the healthy guts. So amplitude is not the reason that we're seeing more on-off cells. Could it be that cells in injured guts are activating Notch at faster rates? Well, to look at this, Erens took her single cell traces, determined where in the traces Notch was turning on, in other words, where GFP fluorescence was increasing, and measured the slopes. And indeed, she finds that on rates in injured guts are significantly faster than the on rates in healthy guts. She then does the same analysis for Notch off and finds that just like the on rates, the off rates are also significantly faster. So what's speeding up these Notch dynamics? Well, we had a great candidate, the JackSTAT signaling pathway. In many stem cell-based organs, JackSTAT serves as an injury signal. And the fly gut is one of these organs. Here in these whole mount images, you can see how this GFP stat reporter lights up when the gut gets injured. Work from many labs has shown that activation of JackSTAT in stem cell is responsible for accelerating stem cell divisions during injury. Damaged enterocytes release a cytokine called unpaired 3. This cytokine binds to its receptor domeless on stem cells, which then signals to JackSTAT. And this JackSTAT signal activates these stem cells to divide 10 times or more faster than homeostasis in order to replace all these damaged cells. Now, Erens asked, is JackSTAT also responsible for accelerating Notch signaling in stem cell daughters? To get at this, she genetically activates JackSTAT in guts that aren't injured and looks to see whether this is sufficient to alter Notch kinetics. Now, these are the movies I've shown you before, a healthy gut and an injured gut. And here's a movie of an uninjured gut. It's never seen bleomycin. But the stem cells and enteroblasts in this gut express an activated allele of Jack. And what's super interesting is how this gut on the right shows some of the same hallmarks of injury. There's enhanced cell motility, the progenitor cell nuclei are really big, but this gut hasn't experienced any damaging agents. Now, what about on rates? Well, when Erens measures the rates of Notch on in single cell traces, she finds that cells that are activated Jack are turning on Notch incredibly fast, faster even than cells in guts that have been damaged by bleomycin. Yet the cells over here on the right haven't been damaged. So JackSTAT is sufficient to accelerate Notch on. Well, what about Notch off? Well, interestingly, Jack is also sufficient to accelerate Notch off without injury. And we think the ability of this one JackSTAT signal to accelerate both Notch on and Notch off suggests that Notch is initiating some sort of negative feedback during differentiation to eventually silence itself. And here, I don't have the data to show you for time, but Erens gets a similar result also when she inhibits JackSTAT in guts that have been injured, both in terms of Notch on and Notch off. So to summarize, in healthy guts, it's a sunny day. Stem cells divide slow and daughter cells differentiate slow. They can take their sweet time. But when injury hits and the organ needs an all hands on deck emergency response, we've known for a while now that part of this response is faster stem cell divisions in injury. And what I've shown you is that faster daughter differentiation is a part of this as well. In injury, cells move faster, both through commitment, Notch on, and through maturation, Notch off. And what's really interesting to us is how the stem cells and differentiating cells are both responding to the same molecular signal, cytokines that are released from damaged cells that activate JackSTAT in these two cell states. We think this is really elegant because by using the same signal to control different types of cellular responses in injury, this might help to coordinate repair at the tissue level so that the organ can more rapidly rebuild. Well, in my last couple minutes, I want to link these findings on cell differentiation to quality control. The fact that cells can differentiate faster raises what I think is a really interesting question, which is, if a cell can differentiate fast, why doesn't it differentiate fast all the time? And faster should be better, right? Well, we might imagine that there's a trade-off between speed and quality. And this trade-off is captured in my favorite episode of the iconic I Love Lucy TV show. Lucy and Ethel are working in a chocolate factory wrapping chocolates, and it all goes well at first. But then when the speed of the production line ramps up, the quality of the wrapping goes down. And so I'll end my talk by posing a question. When cells differentiate fast, what sort of quality might they have to give up? And finally, I'll close. Here's now Dr. Erens Guts, who single-handedly drove all of the work that I've talked about today. This is her giving her defense just last week. And this is our lab's graphical t-shirt abstract that we use for our Zoom background for her defense. Thank you.